Great. Thank you all for coming and welcome to this Weill Cornell Cardiometabolic Symposium. Before I start, I would like to thank the Gulf Heart Association that gave us a chance to do this symposium, and I would like to call His Excellency Dr. Hajar to the symposium.
Good evening. On behalf of Gulf Heart Association, I came to thank Weill Cornell Qatar for their participation with our organization to make this conference successful, and I hope they will join us in the future conferences also. And thank you.
Thank you. Yes. Thank you, Dr. Hajar, for the opportunity to take part in this conference and for including us in this wonderful event. Now to the science. Thank you.
Thank you again, Your Excellency, and we are very happy, very glad to collaborate with you all the time. So, first of all, I would like to apologize on behalf of my colleagues, Dr. Jassim Swady and Dr. Nidal Asad, who both got, unfortunately, some family emergencies and they couldn't make it. The idea behind doing a cardiometabolic symposium during the Gulf Heart Association just came from the fact about the burden of diabetes in the region and the burden that diabetes provokes to cardiovascular diseases. I had the pleasure to work with my colleagues in here through the several cohorts, and a lot of them have been presented in here. In fact, when we looked at the prevalence of diabetes in the GCC countries, it's almost 20%, which is really higher and double than other countries. And when we look at the burden of diabetes in cardiac diseases, up to 60% of patients with heart failure and with acute myocardial infarction have diabetes. And if you want to compare it to other countries, it ranks between 35 to 40%. And when we look at those patients with diabetes, they have worse outcome, increased mortality, whether in hospital or on the long term. I will give the talk right now to my colleague, Professor Khalid Mashaka. We'll talk about the Weill Cornell's mission in Doha, and then later on we will go to talk about the pathophysiology of diabetes, some clinical data, what drugs are effective and are not in diabetic cardiovascular disease, a brief epigenetic talk, and we'll finally finish with our colleagues from Lebanon, who will give us some clue about the cardiovascular research in cardiac and disease in the Middle East. So I'm glad to introduce Professor Khalid Mashaka, who is also the Associate Dean for Research at Weill Cornell. Dr. Mashaka is an internationally renowned scientist in calcium signaling and has received several intramural grants, extramural from the NIH and the Qatar Foundation. So in his role, he oversees the research at Weill Cornell in Doha, whether from a scientific point of view, administrative point of view, and of course the financial point of view. So please, Khalid.
Thank you, Sharbel. Can you guys hear me okay if I walk a little bit around? Everybody? Yes. Okay, so thank you, Sharbel. I'm representing Dean Sheikh in this talk, who sends his regard. Unfortunately, he couldn't be with us. He's traveling. I want to thank Dr. Hajar and the organizing committee for including us in this event, and I'm sure the conference has been quite successful to date. So what I thought I'd do, I'm going to take very little time, a few minutes, just tell you a little bit about the research effort that's been ongoing at Cornell over the past decade. So Cornell Research started about 9 years ago with a very specific mission, which is stated up here. I hope everybody can read it. The goal was, in collaboration with Qatar Foundation, to build a center for research that spans the spectrum from basic to translational to clinical research. We wanted to build a very robust infrastructure of exceptional scientists, and really the goal was to target diseases that are critical for the nation of Qatar and improve them on the long term, including, importantly, cardiovascular disease, diabetes, and obesity as well. So the way we started the effort was to build a three-tier structure that's illustrated here. We put a lot of effort into research administration. Unfortunately, the pointer is not working, so I'll walk you through it. The blue bar, which is research administration, establishing the infrastructure to support the research enterprise from an office of grants and contracts, a business office. Of course, all the regulatory infrastructure that supports research from IRB, IACOC, IBC to support the research, a lab management component, and a training and outreach component that I'll talk a little bit more about. And also from the beginning of the inception of the program, we put a lot of effort into building centralized core labs. That's the green bar in the middle. And the idea was, if we're going to target diseases that are critical metabolically, we have to understand at the whole organismal level what are the changes happening. So we have a genomics core, proteomics core, I'll talk more about it. And also cores that support imaging and animal experiments, like an animal house. And clinical cores like biostatistics and clinical research support. This whole effort is to support faculty efforts to do the research, to be able to advance the science, and ultimately, like I said, be able to treat the patients better in Qatar. So very quickly, the cores, what they do, genomics core does a lot of deep sequencing, genotyping, methylation, and expression profiling. And again, the goal is to better understand diseases, especially complex one, where it's not clearly linked to one gene that leads to the disease. Proteomics focuses on quantitative proteomics, post-translational modification, and also lots of clinical proteomics using the SOMA platform, which some of you may be familiar with. Bioinformatics and metabolomics, microRNA core, an imaging core that touches on the latest technologies in imaging, flow cytometry, histology. And the last two cores focus primarily on clinical research, biostatistics core to help physicians analyze their data more efficiently, and the clinical research support core to help with sample storage analysis and building databases that support the research effort as well. The regulatory infrastructure, I won't spend a lot of time there, but certainly to be able to conduct research, you have to have that infrastructure in place. And we touch on all the three areas: biosafety, animal, and the IRB, of course. The IRB works very closely with HMC, where most of the clinical research is happening. So a little bit of numbers about the research effort. This is the growth in personnel in the top graph over the past few years, where we capped at about 200 people in research, and then there's a small decline given the decrease in funding that we've experienced lately in the country. And below is the cumulative extramural research funding that our faculty have been able to garner over the past few years. Importantly, the research effort has been quite successful in terms of publications and citations. This is the number of papers published. We're over 800 papers published to date. The institutional H index of 52 is actually quite impressive. And the total citations to date from the papers published by the faculty is over 16,000 citations, which is quite good. And the papers published by our faculty, a good number of them are in top-tier scientific journals, which is also quite good. There's also a lot of effort on training, both in terms of research personnel and graduate students and medical students, of course. We have a training program that's focused on nationals to really entice them to get engaged in research and be able to learn more about what the full biomedical research spectrum offers. So to date, we've trained 35 nationals. This is actually a residency program, if you will, where the trainees spend 6 months with us as employees, and they train fully in terms of what is research. They actually conduct a research program themselves, so on and so forth. very significant numbers of trainees as research specialists and clinical research coordinators. We've trained or training or graduated about 27 graduate students to date, primarily in collaborations with European institutions at this time. We collaborate nationally on the training front with HMC, CIDRA, and others in terms of training local professionals as well on research and how it's conducted. We're also involved heavily with medical student training, as you would expect. I'd like you to focus on the top graph here, which is quite unusual for a medical school. This is the percent of our graduates that have had a research experience before graduating over the past 4 or 5 years. And basically, every student before graduating engages in research, which is very encouraging for this. But also, they have many opportunities to do research as early as pre-med students during their medical career, as residents, and certainly as faculty when they come back and join us. Many events and seminars, I won't talk about them, conferences, workshops, that also will allow the research community to engage in research, learn about the latest and the greatest, and contribute. So I'll stop here. If there's any burning questions, we might take one question. If not, we'll go to the next talk. Okay. So it's a great pleasure to introduce Dr. Mark Cooper, who joined us from Australia. Mark is a professor of medicine at Monash, and he's been the chief scientific officer for the Baker Heart and Diabetes Institute. He's internationally recognized for his research in diabetes, both at the basic front and clinical front. And he's the recipient of the Officer of the Order of Australia, which is quite impressive. And today he'll talk to us about the latest effort he's done on the basic front. Mark.
Thank you. Is it working? Yeah, that's the slides. Okay. Well, thank you very much. Thank you very much for that kind invitation. And it's a great pleasure to talk about diabetes and cardiovascular disease. And my task is to give you a bit of a background about basic research in the area. And I've picked a particular area, particularly the role of oxidative stress with some involvement in inflammation. And as you would be aware, the major burden of diabetes is its complications. And indeed, diabetes, the major cause of mortality, is cardiovascular disease. And increasingly, as many of the other risk factors for cardiovascular disease are doing very well, lipids, hypertension, reduction in smoking, unfortunately, the epidemic of diabetes means that cardiovascular disease will not disappear over the next few decades. And with the Gulf region being one of the major hotspots for type 2 diabetes, we are really facing a major epidemic here, not only of diabetes, but cardiovascular disease, potentially blindness, and kidney disease. So, big question that's always asked and is not very well appreciated, particularly by clinicians, is that the effect of glucose control alone on cardiovascular complications has been remarkably disappointing. And although glucose is the defining diagnostic criterion for diabetes, surprisingly, till very recently, reducing glucose per se has been a very disappointing approach for reducing cardiovascular disease. We've seen better links to renal disease and to eye disease, but even there, reduction in glucose is not really translated as one would have hoped to a reduction in the burden of diabetes and its complications. So we need to think more in a more advanced way. Why is it that glucose control alone has been so disappointing with respect to cardiovascular disease? And although in the last 2 years we've had some seminal studies, which we'll hear about later in the symposium, showing that certain glucose-lowering agents might confer cardiovascular benefit, overall there is an enormous gap in managing diabetes. And we know that glucose itself generates a whole range of other products. And what we believe is it's these products generated by hyperglycemia, or indeed can be generated from other sources, which we call toxic metabolites, which we think play a critical role in the development of complications. And maybe where glucose control alone is not that impressive, maybe if we could address these toxic metabolites, we could deliver a reduction in the burden of complications. Now, there are a whole range of these toxic metabolites, and I'm going to focus in particular on one particular area, and this is reactive oxygen species. So glucose and indeed lipids can generate these reactive oxygen species, but often it's not a direct production of the reactive oxygen species, but it's through intermediates known as reactive dicarbonyls, of which the classical one is methyl glyoxyl. And then these can generate reactive oxygen species, or they can generate what we call advanced glycated end products. And our research over the last 20 years has focused on approaches where we could suppress either the generation or the deleterious biological impact of these AGEs, these reactive dicarbonyls, or these reactive oxygen species. And these reactive oxygen species and these other intermediates trigger a whole range of both functional, structural, genetic, molecular, cellular functions which will ultimately lead to diabetic complications. And although I won't talk about this in detail, suppression of many of these intermediates we've shown both in vitro, in preclinical studies, and more recently clinically, can lead to a reduction in these events which promote complications. So if we look here clinically, and these are data from the ADVANCE study that are as yet unpublished, despite more than 100 papers from this multinational study where Michelle Maher, one of the other speakers, was also involved, what we've looked at is a particular oxidative metabolite, and you can see that when we measure that in the serum of these type 2 diabetic patients at risk of cardiovascular disease, we see that as you have higher levels of this particular oxidative stress product, you see an increased risk in mortality, which is primarily related in the right panel to cardiovascular death. So there is a very close clinical association between markers of oxidative stress and increased cardiovascular events. Now, this is only an association, and what our work has done is identify that there is clearly a pathogenic link between these reactive oxygen species and the susceptibility to diabetic cardiovascular disease. So there are many sources of reactive oxygen species in diabetes, and the classical work is related to mitochondria, which produce reactive oxygen species, but our own group has studied all of these pathways, and in particular we believe that one particular family of enzymes, NADPH oxidase, probably plays the major role in promoting oxidative stress in diabetes. And the classic story of increase in oxidative stress was really identified in the early, around 2000, by seminal work by Michael Brownlee, published in Nature, where he showed in vitro that endothelial cells will have increased reactive oxygen species generation in the setting of hyperglycemia. This was shown originally in endothelial cells, and subsequently in this lower slide you can see these are isolated glomeruli from the kidney, and there is a stain here for superoxide, which is markedly upregulated in diabetic glomeruli. And he showed subsequently that indeed if you take out the mitochondria from cells, the high glucose induced generation of reactive oxygen species was prevented if you had no mitochondria, or in the right, if you blocked superoxide production by overexpressing products, UCP1 or manganese superoxide dismutase, you could prevent this increase in superoxide production. Now, we've actually been able to show that this change is occurring throughout the body. And here you can see, for example, this is work we've done in the kidney, where we've been able to show that there is evidence of respiratory electron transport chain dysfunction in diabetes associated with increased mitochondrial reactive oxygen species. And indeed, if you do electron microscopy on the diabetic kidney, you will see abnormalities in the mitochondria, which probably are linked to this increase in reactive oxygen species. So we believe that there is in vivo evidence in humans, as well as in our preclinical models, that there is mitochondrial dysfunction in diabetes, in the heart, in the kidney, and in the vasculature. And we believe that this is a very important phenomenon, and that the mitochondria are working as more than just the powerhouse in the kidney or in the heart, but they are also playing a key signaling role, which might be important for calcium handling, redox signaling, and apoptosis. And endothelial cells have limited energy through mitochondria, so probably these changes in the mitochondria in the endothelial cell are more related not to bioenergetics, but to some of these signaling abnormalities that we see in diabetes. So what we believe happens, and it's a recent review that was performed by our group, which shows that what you have in diabetes is an abnormality in the signaling and in superoxide production, so you end up with increased mitochondrial superoxide. So this is thought to be a major abnormality, but has turned out to be very difficult to target from a clinical point of view. But as I mentioned earlier, we believe that a more targetable source of superoxide that could potentially be more important in the diabetic setting is this family of enzymes known as NADPH oxidase. Now, there are a range of these isoforms, and they play a critical role in the whole situation with respect to diabetes, but they also interact with a whole range of other pathways which might be important in other diabetic complications, such as the polyol pathway. But importantly, there are many factors in the diabetic situation that activate this enzyme, NADPH oxidase. And what we see is not only increased expression of this enzyme, but indeed increased activity. As I mentioned earlier in the previous slide, the role of NADH that is generated, this is an increased substrate for the enzyme, but all these factors on the left in that box, such as angiotensin II, changes in redox state, stress and strain, as we would see with hypertension, can activate this family of enzymes. And we know there are a whole range of these enzymes, isoforms. There's NOX1, which is particularly important in the vasculature, NOX2, which is important in immune cells, neutrophils. It's the superoxide generated by NOX2 that fights infection. There's NOX4, which is known as Renox, is primarily in the kidney. And then you have NOX5, which has been an underappreciated isoform, which is only found in humans and is less well studied, but we believe could potentially be the major source of superoxide in the human context. So we've tried to explore the role of these various isoforms, and I'm going to show you a lot of data with NOX1, because NOX1 is the major isoform that is generating superoxide in the vasculature. It has about 60% sequence homology with the classical NOX, the NOX2 that's in the neutrophils that fights infection. And NOX1 expression has been clearly shown to be activated by all those stimuli that I mentioned earlier that are increased in the diabetic situation. So this is, for example, showing you some data where angiotensin II, this is a non-diabetic context, has been infused, and you can see an increase in superoxide staining when you infuse angiotensin II. This is on the left side. However, if the animal has NOX1 deleted, this increase in superoxide does not occur. So this suggests that NOx1 could be the powerful major source of superoxide in large vessels. So to study this, we generated unique knockout animals where what we did is we knocked out the NOx1, the NOx2, and the NOx4 isoform and put the animals on an atherosclerosis-prone background, the classic ApoE background. And then we induced diabetes in these animals. And you can see here in the diabetic animal, we see an increase in generation of a superoxide in the diabetic wild-type animals. In contrast, just as we saw with the infusion of angiotensin II, we saw no increase in superoxide in the diabetic NOx1 knockout animals, consistent that both hypertension and diabetes are probably generating superoxide in large vessels through NOx1. And this indeed translated to reduced atherosclerosis. So if you look here on the left, you see the NOx1 replete animals, and there is some evidence of atherosclerosis, and the brown staining on the right is showing it in a diabetic animal, and there's a lot of staining, particularly there's macrophages and early foam cell formation, and this does not occur in the right lower panel where NOx1 has been deleted. And you can see we can measure the plaque area in these animals. Diabetic animals have a 2 to 3-fold increase in plaque, and this is not seen in NOx1 deleted animals. By contrast with NOx2, we have a different situation. So NOx2 is known as GP91-FOX. It's the classical NOx. It's the one that produces superoxide and fights bacteria. And indeed, if you induce diabetes in NOx2 knockout animals, they will all die. And they all die because of gram-negative septicemia, because they are unable to fight the infection that occurs in the context of high glucose. By contrast, in the absence of diabetes, there is no mortality. This emphasizes that if we're going to generate a NOx inhibitor, it must not inhibit NOx2. If we look at NOx4, this is constitutively active. It's primarily in the kidney, although it is seen in other cell populations, and is also activated by the same stimuli as NOx1. And if we look here in contrast to the NOx1 knockout, NOx4 knockout animals actually have increased atherosclerosis. And this emphasized that although these enzymes have quite similar homology, they probably have different biological roles depending on their location. But interestingly, the renal disease, this was known as Renox, you see this marked increase in albuminuria in the diabetic animals is not seen in the NOx4 knockout. So in summary, the NOx1 deletion is linked to atherosclerosis, NOx2 is linked to infection, and NOx4 is linked to the kidney disease. I won't show you the data, but we've also looked in the retina, and in the eye, it's NOx1, very similar to the vasculature. So if we look, you can see here, now we have followed NOx4 in these animals. We see, interestingly, that there is an increase in NOx4 in the aorta initially, but over time, you end up with a depletion of this enzyme. Interestingly, as I showed earlier, there is also an increase in atherosclerosis. So NOx4 alone, if we delete it, there is the risk that although it will be renoprotective, we could be promoting atherosclerosis. Finally, I want to talk about the last NOx isoform, NOx5. It's present in humans and primates. It's also present in the rabbit, but not in rats or mice. So it has been much more difficult to study. It is calcium sensitive, and it does not require many of the other subunits that the other NOx enzymes require. This is some unpublished data where, because NOx5 is not normally present in a rodent, we've had to make a transgenic animal. And here you can see we've overexpressed NOx5 in the lower animal, and you can see a marked increase in atherosclerosis. So NOx5 is a very important generator of superoxide, and even in the absence of diabetes, an increase in NOx5 will be pro-atherogenic. So what do we want to do? We want to inhibit NOx1 for the vasculature. We want to inhibit NOx4 for the kidney, and potentially, because we don't know in rodents, we want to block NOx5 as well. And we've been very fortunate to have access to a compound called GKT-137831, which inhibits NOx1, but not NOx2, because this would promote infection, as well as NOx4 and NOx5. And we have given this drug to animals, all the various different animal models, and you can see here that when we gave diabetic animals this GKT compound, we ended up with reduced atherosclerosis, as well as reduced oxidative stress within the vasculature as measured by nitrotyrosine staining. And indeed, this just shows you the effect on atherosclerosis with this GKT compound, and it's very impressive. And if you look on the right, you can see the ApoE diabetic. This is staining through the coronary sinus, a lot of staining for plaque, which is reduced but not totally prevented by this compound. And we have recently been funded to study a trial in patients at risk of cardiovascular disease with type 1 diabetes, and we're going to look not only at the vasculature, but because this also blocks NOx4, albuminuria. So I've mentioned to you NADPH oxidase, but there are other sources of oxidative stress in the diabetic situation. For example, NO synthase in the high glucose environment becomes uncoupled, and it changes to produce superoxide. And you can see here in the endothelial cell in the diabetic situation, you see the uncoupling, and you end up generating, instead of generating normally L-citrulline, you end up with superoxide. And indeed, this has been shown primarily preclinically to occur in the diabetic context. And this just shows you that you can block this phenomenon by inhibiting NO. And this is just here that you've got the increase in superoxide in the diabetic situation. And if you give a NOS inhibitor, as well as some of these other molecules that I won't go through in detail, it's been shown very early on that all these antioxidant approaches can reduce superoxide, emphasizing the multiple sources of superoxide in the diabetic situation. eNOS activity is actually under the control of caveolin-1, and a very bright PhD student from our group showed quite a few years ago that by giving a blocker of the caveolin pathway, by blocking the ability of eNOS to interact with caveolin, by giving a mutant form of caveolin, cavnoxin, you can reduce plaque. That's on the right. This has been shown in both a type 1 and a type 2 diabetic model, and it's associated with a reduction in staining for superoxide in the vasculature, further emphasizing this as a major source of superoxide in the diabetic setting. Lipoxygenase is also an important source. We have recent data where we've also explored this. This is very important. Some of the eicosanoids play a key role here, and they are also potentially targetable in diabetes. But it's important to appreciate that I've talked mostly about the generation of superoxide, but it's important to also appreciate that there is also a way to get rid of superoxide, and this is the antioxidant defense. There are a whole range of enzymes that will turn superoxide into water, and in diabetes there is impaired antioxidant defense. And for example, one of the key enzymes is GPX1, and we were able to show that in a GPX1 knockout, where you further impair the antioxidant defense, you can see here, you see an increase in the coronary sinus of atherosclerosis and increased plaque area. Now you can reverse this problem with GPX1 by giving a GPX1 agonist. And indeed, epsilon, which is on the right, is an antioxidant defense molecule, because you give epsilon, you replete GPX1, and this leads to a reduction in atherosclerosis. So it's very important to remember not only to reduce superoxide generation, but to promote its degradation to water. Now, increasingly it's appreciated that there are other forms of antioxidant defense involving this NRF2 KEAP1 pathway, and you can actually, by blocking its degradation, you can promote further defense in the antioxidant situation, because NRF2 is clearly regulating a whole range of antioxidant cytoprotective genes. It also plays a critical role in NF-kappa B activation, which is the master switch for inflammation, and so any of these targets will also potentially have anti-inflammatory actions. And we've been fortunate, we've been able to use an NRF2 agonist and shown that the increase in plaque in the diabetic animals, the red column, can be reduced by this NRF2 agonist. Now it's interesting to appreciate that as you go, these numbers on the bottom are the dose, and you can see as you increase the dose, suddenly you start to see a reversal of the benefit. And we've been able to show that in vitro, increasing this NRF2 agonist, it changes from being anti-inflammatory to pro-inflammatory and increases NF-kappa B activation, increases chemokines such as MCP1 to promote inflammation and atherosclerosis. So this is a very difficult area because we're trying to balance the generation of superoxide, influence antioxidant defense, and this has turned out to be a bit of a nightmare for the pharmaceutical industry because there is a very small window, therapeutic window, and we all know drug companies don't want drugs that if you have to increase the dose, you end up with either an adverse effect or indeed side effects. So this has turned out to be a very difficult area, and one of the classic NRF2 agonists is a drug called bardoxolone, which turned out, there was a very nice New England Journal paper showing that when they gave a higher dose of this agent, indeed they did not see the initial benefit on the kidney that they had seen initially at lower dose, and they also saw cardiovascular side effects. So this is an area where people have to research it very carefully from a clinical perspective. So where are we today? Well, I've tried to give you a lot of data, but it all has a central theme. Glucose in a vulnerable cell, such as an endothelial cell, which is unable to turn off glucose transport when it sees high glucose, so the glucose all enters the cell. It makes these reactive oxygen species, and then you either have a problem with defense or you have an increase in production. The problem is that this reactive oxygen species itself, when it's generated, will influence these pathways and perpetuate and amplify the pathway. And this is known as the kindling hypothesis, and that's why it's very important either to hit glucose or early on upstream reactive oxygen species to prevent this amplification. So in diabetic complications, what we're trying to do is if we can't reduce glucose, at least reduce these generated toxins, superoxide, methylglyoxal, AGEs, activation of enzymes, the NOx enzymes, protein kinase C. And by doing this, I think we can reduce the inflammation, the oxidative stress, and many of the other pathological processes that promote diabetic complications. And I think clinically we're seeing new drugs such as the GLP-1 agonists, SGLT2 inhibitors, which are probably in part inhibiting some of these pathways independent of lowering glucose. So I'd like to just stop, welcome you all to Australia. Australia is far away from nearly everywhere in the world, but fortunately Doha has a direct flight to Australia, so it's one of the few places in the world you can get to Australia without a stop. And this is our new Department of Diabetes at Monash University. Thanks very much.
Thank you, Mark, for a beautiful talk. We have time for a few questions. Maybe I'll start with one. The GKT compound seems like a perfect drug. Do you notice any effects on other complications of diabetes, like neuropathies and things like that? Is ROS central for other things other than atherosclerosis?
So the GKT compound's been shown preclinically to be very good in retinopathy, nephropathy, and in macrovascular disease. We don't know very much about cardiac function, and recently when I was in Lebanon, I was told that they have some early evidence that it might be useful in experimental neuropathy. So I think that's very important. In our clinical trial, we will do some measures of other complications, but one of the stumbling blocks we might hear about later in the session, diabetic complications take years to develop. So even a short trial of 6 to 12 months, you're very limited in what you're going to look for, and that's why we've chosen albuminuria. The other complicating situation is that NOx is suppressed once you have the complications. So you probably have to target people quite early. There has been one short trial of NOx inhibition in very advanced patients, and it didn't work. And that's because there's no NOx enzyme left in the damaged tissue.
Great, thank you. Other questions?
Can I?
Yeah.
Sorry.
Mark.
Oh, we're going to start here and then we'll go.
Okay. So Mark, thank you for your talk. So in 2001, when Michael Brownlee published that fantastic paper in Nature, I honestly thought we were going to be out of a job. 20 years later, I'm not out of a job. So what is, because, you know, he beautifully worked out the mechanism, how if you give thiamine, you can switch off all these pathways, and we are where we are in 2018.
Because, I can say it here, I wouldn't say it in New York, he got it wrong. And what he got wrong was that he thought it was all in the mitochondria. Turned out there were many other sources of superoxide which were not targetable through his approach. And most of his experiments were in vitro. But once you go into the animals already, you can see, first of all, it's very hard to target mitochondria. Most drugs won't get in there, so that's very hard. So we've tried with MitoQ, and there's now newer drugs, MitoG. We get some benefit, but nothing like what we see with GKT. And that's why I think, as the Associate Dean said, we think you need to target the NOXs, because NOx is the only enzyme, its job is generating superoxide. All the others, they have other roles, and when they generate superoxide, it's an accident. So constitutively, you need to hit the NOx pathway. So I think he changed the way we thought, but he didn't really nail it in which was the right target. And although I haven't shown it today, when we've looked at the epigenetic pathways which are influenced, those are very much regulated by the Brownlee pathway. So I think diabetic complications are multifactorial, and that's why hitting one thing and the unifying hypothesis was probably simplistic, but at least it was a paradigm shift in the way we were thinking, because till then we were stranded.
Yeah.
Sorry, my answer was a bit long.
Thank you for the talk, Dr. Ehsan Rafi. My question is not basic sciences, because
That's
this is
okay.
quite a complicated subject. But I'm confused with your first slide. Are you trying to say that the tight glucose control has no influence on MI, mortality, and so on? The
Yeah.
very first slide that you showed.
So that's probably overstating it. But if you look at the data for intensive glucose control, like UKPDS, ADVANCE, the effects on cardiovascular endpoints have been pretty disappointing. Then we had all the DPP-4 inhibitors. They lower glucose very well. They saw almost no effect, actually zero effect, on cardiovascular disease. So although epidemiologically glucose is closely linked to macrovascular disease, the clinical interventions till very recently were not very impressive. So although most doctors, including cardiologists, thought glucose control will deliver a lot of reduction in macrovascular disease, it didn't. Why that happened, we're not sure, but it is possible that there were adverse effects the way we were reducing glucose. Maybe the development of hypoglycemia counterbalances the benefits of glucose lowering, because in the ACCORD study we saw where there was a lot of hypoglycemia. Indeed, there was increased sudden death and increased cardiovascular death. But the newer agents like the GLP-1 agonists and SGLT2 inhibitors, which we might hear more about in this session, seem to have delivered something in this area, indicating that there is something either that we were lowering glucose the wrong way, or there is something glucose independent with these agents that will confer cardiovascular protection. So giving this talk 2 years ago, I would have been very pessimistic. I think the last 2 years with all those New England papers in both cardiovascular and renal, I think we can say that it's shifted. The paradigm has shifted, and we can improve the outlook of glucose lowering, but it's probably the way we lower glucose, just like it's the way you lower blood pressure that might be beneficial for heart failure or renal disease. I think we can extend that to glucose. Unlike lipids, where it just seems if you lower cholesterol, you see benefits. It doesn't matter which way you lower it. I don't think we can say that about glucose.
Take one more quick question, quick answer maybe.
Sorry.
It is a quick question.
Yeah.
Just the role of glycemic memory. Do you think that the reactive oxygen species is the major upstream pathway for glycemic memory, and then therefore diabetes-induced vascular complications?
So I'm not sure I know that this topic may be addressed by another speaker. In our paper originally in 2008, we were able to show that our model of hyperglycemic memory that we did some work with Brownlee, we were able to show that the memory was ROS dependent. And that in actual fact, one of the reasons ROS might be important is that once the glucose goes down, the ROS often stay up. So it's a persistent mediator of previous exposure to glucose.
Great. Let's thank Mark again for a beautiful talk. So our next speaker is Professor Michel Mar. Michel comes from France. He's a professor of endocrinology and diabetes at Paris 7 University, which has recently joined the Sorbonne. He's also the current president of the Francophone Foundation for Diabetes and the previous president of the French Society for Diabetes. Michel will talk to us today about diabetes and its complication on cardiovascular disorders. He's also led many large-scale clinical trials in that area. Michel.
Thank you, Mr. Chairman, and thank you very much to all of you for this invitation to the Gulf Heart Association and to the Weill Cornell University, and a special thanks to my good friend, Dr. Charbel Abi Khalil. So, these are my several conflicts of interest, which are in fact interests common to other industrials. Just to remind you that the definitions of diabetes rest on a certain level of glucose that makes microvascular disease in addition to macro or cardiovascular disease. This has been obtained from prospective follow-up studies, especially those of the Pima Indians in Arizona over 15 years of follow-up. So the zone of impaired glucose tolerance provides cardiovascular risk, while true diabetes provides both micro and macro macrovascular risk. So this is the classical picture of type 2 diabetes in the old times. And this is derived from a study by William Cullen, the founder of the Framingham Heart Study, where you see that the relative risk for cardiovascular disease is multiplied by 2 to 6 according to your sex if you are with diabetes compared to those who have no diabetes. And type 2 diabetes is also an aggregation of cardiovascular risk, aggregation of high blood lipid, dyslipidemia, also tobacco consumption and weight excess that would lead to non-glycemic intervention that in fact have been very effective to reduce the absolute risk. But it is a multiplier for each of the cardiovascular risk factors, blood pressure, lipids, and tobacco. But if high glucose is a marker for cardiovascular risk, then intervention on high glucose is a valuable option. And so you will test it through clinical trials. And this idea is not new. It has been elaborated by US diabetologists in the 1960s, and the first trial has been published in 1970. It is a university group diabetes program, but unfortunately, just believe me because I have no arrow in order to show you, but in fact, the patients were randomized either to sulfonylurea, biguanides, insulin, or no treatment, or just diet and exercise, and you see that there was an increase in the risk of cardiovascular death due to sulfonylurea, which of course made a lot of debate. And this is the reason why, some years later, the UK prospective diabetes study, the UK PDS, has been designed. So this is a 20-year interventional trial from 1977 to 1997. More than 5,000 patients with newly diagnosed type 2 diabetes were recruited in between 1977 and 1991 with a median follow-up of 10 years. And there was a post-trial monitoring for 10 years from 1997 to 2007. So a mean overall follow-up of 17 years ranging from 16 to 30 years. And this is a summary of this trial. There was a 12% relative risk reduction from any diabetes-related endpoint, which was entirely attributed to the relative risk reduction for microvascular disease, 25 relative risk reduction, no significant impact on all-cause mortality, no significant impact on myocardial infarction, a 16% risk reduction, but with a p-value of 5.2. So if you are a strict statistician, you say this is not significant. But look at the stroke event and you see it would be a little bit dangerous to reduce blood glucose if you want to avoid stroke. Fortunately, there was a follow-up of this study, and you see that 10 years after completion of the study, while both groups had joined to the same HbA1c level, there was a benefit that appeared on total mortality, and also, I must say, on myocardial infarction, because the hazard ratio was the same, but the number of cases were more important, so the p-value was downloaded below 5%. So as mentioned by Mark Cooper previously, there were several trials that indeed tried to demonstrate that reducing blood glucose would reduce the cardiovascular risk. And I'll illustrate with the advanced study already mentioned. Yes? Thank you. I would like to go back. Yes. The primary endpoint was a composite of micro and macrovascular endpoint, and you see that there was a very significant reduction in the microvascular endpoint, but nothing on cardiovascular endpoint. And regarding microvascular endpoint, fortunately, it was very interesting to note that most of the benefit was due to reduction in kidney events and especially the prevention of proteinuria and of end-stage renal disease, which by consequence might influence the vital prognosis on the very long term. In total, a meta-analysis of individual participants to the 4 studies, UKPDS, ACCORD, ADVANCE, and VADT, over 4 years, has been published. It is the control study, and you can see that there was a 9% relative risk reduction for major cardiovascular events, a 15% significant reduction for non-fatal MI, but nothing significant on all-cause mortality and cardiovascular deaths. So, one exception was intervention on patients with overweight in the UKPDS study that were allowed to be randomized to metformin. Metformin is an old drug that has been developed by Dr. Jacques Stern in Casablanca in Morocco at time of French colonization. And you see that from these data obtained on 341 patients, there was a very significant effect of metformin on risk for death and cardiovascular disease, illustrated by risk for myocardial infarction. But interestingly, there was no significant effect on microvascular disease. Now, I alluded to impaired glucose tolerance, dysglycemia, and you have seen that, in fact, the risk for cardiovascular disease starts by blood glucose values that are far below those defining diabetes. And this is a meta-analysis that has been conducted by the group of Estelle Gerstein and Selim Yusuf, where you see that the value that predict cardiovascular events in the general population are indeed much lower than those defining diabetes. So what did they do? They were courageous enough to translate from the prospective observation, epidemiology, to causation. So they manipulated the blood glucose, and this is the ORIGIN trial during which patients with a small diabetes and a high cardiovascular risk were allocated either to conventional treatment or to nighttime insulin glargine in order to reduce fasting blood glucose below 90 milligrams per liter, and this per deciliter, and this was in fact obtained. But you see the results, not a single effect on cardiovascular risk. So, new and old anti-diabetic drugs, I think I've reviewed insulin, biguanide, that is metformin, and sulfonylurea. So since this time, just to remind that insulin has been discovered in the 1920s, metformin and sulfonylurea launched during the 1950s. New drugs were developed, alpha-glucosidase inhibitor, I shall not allude, thiazolidinediones, and incretin mimetics, DPP4 inhibitors, and GLP-1 agonists, and more recently the SGLT2 inhibitors. So, in fact, the classical picture that everything was quiet, you would reduce blood glucose with any of the drugs, was disturbed by a meta-analysis that has been published by Dr. Nissen. Effect of rosiglitazone on the risk for myocardial infarction and death from cardiovascular causes, meaning that in fact the use of rosiglitazone would affect this risk for myocardial infarction and perhaps cardiovascular death. So the FDA has considered this issue and remembered all the old trials, including the first one, and they decided that all new anti-diabetic drugs should demonstrate their neutrality against the cardiovascular risk. So, in fact, it is a little bit disturbing because the classical cardiovascular outcome studies in cardiology, they have to prove the superiority in terms of events, typically the statin outcome study. Here, they were designed, the cardiovascular outcome studies I shall show you were primarily designed to demonstrate non-inferiority against placebo with the possibility to test superiority according to hierarchical procedure. That is, if you satisfy, then you can test. And just a reminder of what you can expect from the two types of incretin mimetics. The GLP-1 agonist, so you inject GLP-1 exogenously and you get high pharmacological levels of GLP-1. While with a DPP-4 inhibitor, you block the catabolism, so you obtain relatively low levels. So the effect on HbA1c, on weight, and on systolic blood pressure are more important with the GLP-1 agonist than with the DPP-4 inhibitor. Both classes are absolutely without risk for hypoglycemia. They are neutral or sometimes favorable for lipids, but of course, do you prefer to be injected every day or to take a pill every day? I think that you would choose the pill, and another reason is that the high dose provoked by GLP-1 agonist may provoke some side effects in about 10% of the population. So I've reviewed all the cardiovascular outcome studies that have been published with DPP-4 inhibitor. They have demonstrated cardiovascular safety but no cardiovascular benefit. The Savoy-Timmy study was the first one with saxagliptin. There was a concern with an increased risk for heart failure, but in fact this has not been confirmed by the other studies, and the meta-analysis has been published showing that probably the risk is about nil. With alogliptin, the EXAMINE study, same results, same neutrality, and with sitagliptin, the TECO study, I will illustrate just to show you that indeed the blue and the red lines superimpose completely regarding the primary outcomes and its components, and also the hospitalization for heart failure, death from any cause, and so that's it. That reduced slightly blood glucose, and that's it. So now, regarding the GLP-1 receptor cardiovascular outcome studies, four studies have been published on to date. One with lixisenatide, the ELIXA study, with liraglutide, the LEADER study, with semaglutide, which is once a week injections drug, the SUSTAIN 6 study, and another once weekly drug, exenatide, the EXCEL study. And I shall try and illustrate the effect on the intermediate phenotype, that is the drop in glycemia or HbA1c, the drop in blood pressure, the drop in weight, and the result on the cardiovascular outcome. And so lixisenatide is not far different from placebo regarding the effect on HbA1c, weight, and blood pressure, and you see that in terms of cardiovascular outcomes, it is very neutral. So I will expand on the most illustrative of the fourth study, which is the LEADER study. And this is a double-blind, placebo-controlled parallel study that lasted minimally 3.5 years, maximally 5 years, with 80% of the patients in secondary prevention setting. So they had an established cardiovascular disease for 80% of them. And the primary outcome was a composite of cardiovascular death, non-fatal MI and non-fatal stroke. And you see the various secondary outcome, very classical, this all-cause death and the component of the cardiovascular outcome, primary outcome. And you see that in fact there was a sudden drop in HbA1c at the very beginning, and thereafter the two arms are narrowing more and more because there was an addition of anti-diabetic drugs more important in the placebo group than in the other. Look at weight. The two lines are very parallel, and there is a 2.3 kilogram difference between the two, which is very important. And if you look at blood pressure, And if you're a specialist of hypertension, you're very happy, because not only the systolic pressure declines, but also the diastolic pressure increases. So the pulse pressure is reduced very significantly, which should be the most important prognostic factor for the outcome of patients with hypertension. And this is the main results. There was a 13% relative risk reduction for the primary outcome, the composite of cardiovascular death, non-fatal myocardial infarction or stroke. And of course, non-inferiority was obtained, but the p-value for superiority was 0.01, so very significant too. And if you look at cardiovascular death, there was a 22% relative risk reduction, also very significant. All-cause death was reduced by 15%, also significant. And hospitalization for heart failure was reduced 13%, but not significantly, but not on the bad side. And finally, for the maniacs of proteinuria or microalbuminuria, like Mark or me, there was a 22% risk reduction of renal events, which was driven primarily by the prevention of proteinuria onset. There was a reduction of new cases of end-stage renal disease, but this was not significant. SUSTAIN 6, I have summarized too much perhaps, but nonetheless the results are very simple, very plausible. This was a short study. The impact of semaglutide on HbA1c, weight and blood pressure is even more important than with liraglutide, and so the primary outcome, which was exactly the same, was very significantly reduced with each of its components, and interestingly there was a reduction of stroke. So recently, once-weekly exenatide study has been published, and you see that the lines are curious, but so it is. There was a reduction of HbA1c by 0.5%. The systolic blood pressure and the diastolic blood pressure were reduced more modestly than in the two previous studies, and the weight loss was also relatively moderate. And they were very unlucky, because in fact there was a non-significant reduction of the same primary outcome with a p-value of 0.06. So the rigorous statistician will say that it is not significant. And you see that death from any cause was significantly reduced. You cannot see, but I tell it to you. The risk for death from cardiovascular disease was not significantly reduced, and there was no effect on hospitalization for heart failure. So, conclusion, among patients with type 2 diabetes with or without previous cardiovascular disease, the incidence of major adverse cardiovascular events did not differ significantly between patients who received exenatide and those who received placebo. Now I'll skip to the SGLT2 inhibitors classes. So the plasma glucose and the plasma sodium are filtered through the glomeruli, and about 70 to 75% of the sodium is reabsorbed at the levels of the proximal tubule, and this is a reabsorption which is coupled with the reabsorption of glucose. And so there is a physiological phenomenon which is called tubuloglomerular feedback. So the amount of sodium that reaches the macula densa will regulate the preglomerular resistances. And high glucose disturbs, there's a disturbance, an impairment of the tubuloglomerular feedback by glucose. And the dose response is not clear, and this is a matter of investigation now in our group. But interestingly, if you reabsorb elevated glucose, then you will reabsorb more sodium, and nonetheless, the afferent arterial will be less vasoconstricted and more vasodilated. So, Dick Cherney has shown for the first time in type 1 diabetic patients that if you give an inhibitor of the SGLT2, then you will increase both the glycosuria and the natriuresis, and so you will restore the tubuloglomerular feedback mechanism. So let's go to the cardiovascular outcome studies, and the first paper has been published, the EMPEROR study in 2015, and you see here the primary outcome results with a 14% relative risk reduction for cardiovascular death or non-fatal MI or stroke. If you look back, you see that the risk reduction for death from cardiovascular cause was 38%, which is quite high. Death from any cause was reduced by 32%, and hospitalization for heart failure was reduced by 35%, supporting the view that the benefit, total of the primary outcome benefit, was mostly due to prevention of heart failure. Now, if you're a fan of diabetic nephropathy, you will see that this was also very beneficial to all aspects of kidney disease, and on this plot you can see that even the number of end-stage renal disease has been reduced significantly. So, very interesting for the outcome in terms of renal outcomes. And more recently, the CANVAS study has been published with results that are consistent with those of the EMPEROR study. You can see from the left, the top panel, that there was a significant reduction in glycated hemoglobin that reduced with time, a significant reduction in body weight, and systolic and diastolic blood pressure were reduced too. And here is the result, a 14% relative risk reduction for the composite, which makes the primary outcome, and on the also non-significant reduction in death from cardiovascular disease, or in non-fatal stroke, or in non-fatal MI, but the composite is significant. Hospitalization for heart failure, there was a 33 risk reduction, very significant and very consistent with EMPEROR. Death from any cause was hardly significant. Regarding albuminuria, the progression was also reduced by 29%, and a composite of 40% reduction in GFR. Requirement for renal therapy or death from renal cause was also reduced by 40%. So in conclusion, in two trials involved, I did not detail, but the design of the CANVAS study is a little bit complicated even for specialists of trials. Anyway, in two trials involving patients with type 2 diabetes and elevated risk of cardiovascular disease, patients treated with canagliflozin had a lower risk of cardiovascular event than those who received placebo, but a greater risk of amputation primarily at the level of the 2 or metatarsal. So there's an issue with amputation that shall be deciphered along with time. So what are the mechanism of action of GLP-1 agonist and SGLT2, and maybe that they may be additive? Should we use both GLP-1 plus and SGLT2 inhibitor? So both reduce HbA1c, blood pressure, and weight, but on the side of the GLP-1 agonist, there's a reduction of biomarkers of inflammation. This has been alluded by Mark, and in fact, I have no time to present all the data, but frankly, this is a good pathway to account for an additional benefit. Also, you will find by histochemistry some receptors to GLP-1 at the levels of the juxtaglomerular cells that secrete renin. So maybe that it can affect the activity of the renin-angiotensin system. On the other side, probably that a sizable or the major component of the benefit due to SGLT2 inhibitor is extracellular volume reduction, very consistent with its effect on risk for heart failure and kidney disease. So a study has been published, yes, with 3 arms, one with an SGLT2 inhibitor, one with a GLP-1 agonist, and one with the combination of the two. And you see the results on the biomarkers. There was an additive effect of an HbA1c, an additive effect on weight, and an additive effect on blood pressure. So maybe that this is the solution. So do their anti-hyperglycemic effect contribute to the cardiovascular benefits? Our group has published recently that the concept of cumulative risk, prevention of cardiovascular disease through reduction of glycemic exposure. And you will see the main slide. If you plot all the clinical trials on glucose control, and if you put into the x-axis the glycemic exposure, which is the drop in HbA1c multiplied by the number of years, and if you put the MACE hazard ratio on the y-axis, you see that there's a wonderful straight linear relationship. And you see now the confidence intervals of this regression line, and you can see the 3 studies, the LEADER trial, the SUSTAIN-6 trial, and the EMPERATE trial, and you see that their confidence intervals superimpose with those of the regression line, supporting the view that in fact there are anti-diabetic drugs and they can read the contribution or the benefit of the reduction of HbA1c may be significant. Thank you for your attention.
If there is any burning question for Professor Moore, we can take maybe one. Okay, sure. Okay, please go. So that's a question from Palestine coming through blog, I guess, or something.
Yeah, they are actually watching us live streaming.
We're live to the world.
Yeah.
That's wonderful.
But they send it by WhatsApp from my colleague, Dr. Mohammed Zughbar. He is a consultant endocrine in Gaza, Palestine. His question in LEADER trial, why DPP-4 inhibitors were in exclusion criteria and why not to be compared with. And the second part of the question, in explanation of that, it reduces the cardiovascular death and mortality, but without affecting the non-fatal MI or non-fatal stroke, although we know that most mortality will be through these 2 complications.
Regarding the first question, in fact, yes, of course, if you act on the same mechanism of action, that is the incretin effect, the DPP-4 were excluded, but in fact, some investigators and patients have violated, and it has been shown and analyzed that in fact the benefit was the same with or without DPP-4 inhibitors on top of the trial. And regarding the second question, the primary outcome is a composite. So in one study you will find an effect on one of the components, and in one study you will find an effect on the other component. But the power has been calculated on the composite. So sometimes it is effective, it is significantly reduced on one component and sometimes on another.
Okay, so thank you.
Okay, great. Thank you again, Mr. Mark. Thank you again. Okay, our next speaker is Dr. Charbel Abi Khalil, who is actually at Cornell. Before coming to Cornell, Charbel was a cardiologist at the INSERM in Paris and at Cornell in New York as well. Since joining us here, he has focused his work on cardiovascular complications of diabetes, both on the clinical, epidemiological, and epigenetic perspective, and I'm sure today he'll give us a summary of that work. Charbel?
Thank you very much. Dr. Mashaka, and again, I'd like to thank the Gulf Heart Association for organizing this big cardiac meeting and for letting do a collaborative work regarding the symposium. So, I know it's at the end of the day and we are pretty much tired, so I'll try to go as quick as possible and as less basic science as possible. So the gentleman here you see smoking a pipe was the first to describe epigenetics in 1942, and he referred to it as the interactions between the genome and the environmental factor that can give rise to differences between cells during embryonic development. And that was based on several observations that stem cells have an identical DNA but have different cell fates. And if we, for example, take right now the hematopoietic stem cells, it can at the end give monocytes, lymphocytes, megakaryocytes, but actually the cell, although they have different functions, they all have the same DNA. The concept of epigenetics has evolved ever since, and now it's widely accepted to name it at the study, or to refer it at the study of heritable changes in gene activity and gene functions that are not due to the changes in the DNA sequence. In other words, these are like the mechanism and the phenomenon that can affect the phenotype of a cell or an organism without affecting the genotype. So epigenetics is found everywhere in the body somehow. As we mentioned, it's essential during embryogenesis. It's also part of the normal physiology and function of the body. And as I'm going to show some examples, it's also implicated in the pathophysiology of diseases such as cardiovascular disease and cancer. A striking example of the epigenetic phenomenon is the monozygotic twins. So the monozygotic twins, as you know, they share the same DNA, but as we encounter them in normal life, they can also have some slight differences. The difference could be like sometimes very obvious, like the hair color. But what is important also, they have difference in the predisposition and in the incidence of non-communicable diseases. That this very nice paper published at PNAS, who assessed the differences, the genetic differences in monozygotic twins. They took twins at the age of 3 years, examined their DNA, and then the same were examined at the age of 50 years old. And during that time, the twins were split, and they were living in different environments and different cities. As you can see, when they were like 3-year-old twins, the epigenetic phenomenon were tagged in some green and some red. I'm afraid I don't have access to the pointer. Right. So when they are 3 years old, most of the color you can see is yellow. It means that they are like overlapping regions. They share the same epigenetic mechanism. But when we look at the 50 years old twins, this has completely changed. So the prevalence, right. So as you can see here, the red and the green regions are much more prevalent, which means differences in epigenetics that has accumulated during the year. And the overlapping one, which is yellow, has significantly decreased. So what are the epigenetic mechanisms known so far? There are 3 of them. The DNA methylation. It is the addition of a methyl suffix at the cytosine base, which is followed by a guanine one. However, sometimes people get confused. We are not talking about this bond that is usually a hydrogen bond between cytosine and guanine, but we're talking about the relation on the same DNA strand. And then we've got the second one, the post-translational modification of histone tails. As you know, the histones are the protein core around which DNA is wrapped, and the N-terminal of these histone tails can have a lot of chemical reactions such as acetylation, phosphorylation, methylation, and so on. And finally, the non-coding RNA. As their name indicate, they do not code for proteins or for polypeptides. However, their function is limited to regulation of the gene function. And they are broadly divided into microRNA and macro, depending if they are 20 to 22 polypeptides, they are micro. And sometimes you can see in a lot of clinical publication, they are pretty much potent biomarkers that can predict sometimes the diseases or even their progression. And actually, in most of the epigenetic studies, it's not like the one or the others, but these epigenetic mechanisms interrelate together and change the gene expression and the gene function. So I'll go briefly into that. Our colleagues in oncology were the first to use epigenetic in practice. And there have been several reports that indeed in cancer patients there is global hypomethylation of the DNA with a region-specific hypermethylation. This is in parallel to changes in the histones. So basically what happens that all of these epigenetic abnormalities accumulate with time and cause a cancer in a cell or at an organ level in response to several factors that are predisposing to cancer. And actually, they are like much more pioneer than us in cardiovascular disease because they're already using these epigenetic drugs, and especially in blood cancers. They have the DNA methylation inhibitors, the histone deacetylase inhibitors. It's very interesting to know how they work because they reactivate the tumor suppressor genes and they restore the normal function. And if you want to go more in depth into that, whoever would like to read about those, the current research there is to correct the epi mutations in people who are somehow at high risk and prevent the cancer. So while the total number of epigenetic publication has been increasing with time, the cardiovascular epigenetics has been lagging behind, and it's less than 10%. It's almost 5% than the total epigenetic publication, which means we should work more and more towards the specialty and not let other colleagues take it. Why are we studying the epigenetics of cardiovascular complication of diabetes? First, it's pretty much important to know the pathophysiology. We cannot offer a treatment to patients, and we cannot go on with new drugs if we don't really know the pathophysiology. Second, as I just mentioned with the microRNA, epigenetic could be used as biomarkers, and hopefully one day we will reach the level of epigenetic drugs. I will go briefly over this because I believe my colleague, Professor Cooper, has already mentioned it. So hyperglycemia can trigger a lot of pathways. There will be increase in reactive oxygen species, which will activate hyperglycemia pathways, and all of them would crosstalk with the DNA through epigenetic mechanism and lead to the inflammation, proliferation, differentiation. All of those factors would finally lead to complications, whether macrovascular or microvascular. So what proof do we have about the implication of diabetes and implication actually of epigenetics when an organ is exposed to hyperglycemia? This very nice paper from the Baker Diabetes and Heart Institute, the author exposed the primary human aortic endothelial cells to hyperglycemia while having a normal glycemic control. They did chromatin immunoprecipitation to study the histone 3. They also did studied the methylation and finally looked at the gene expression. And in fact, they reported that there was a global reduced methylation and increased acetylation. And what is striking about that is the genes that get changed through that. So there are a lot of genes that get upregulated. It means that they are more expressed, and those genes are pro-inflammatory genes, and the ones that are downregulated or less expressed are the ones that usually protect from cardiovascular disease. So other epigenetic mechanisms induced by hyperglycemia, are they reversible or not? Well, in this study, as well as in many studies, apparently it seems that they are not reversible. At least on the short term, one can always argue about the long term. So also from the same team, they exposed bovine aortic endothelial cells to hyperglycemia. However, after the exposition for 16 hours, they were divided into three groups. They were divided into three groups and re-exposed to normoglycemia, 2 days, 4 days, and 6 days. And then they analyzed the subunit P64 of a nuclear factor kappa B, which is known to regulate inflammation and regulate cytokines. And then they realized, as you can see in here, even after going back to normoglycemia for 2, 4, 6 days, the level are still high than the controls. It means that the effects are lasting even after hyperglycemia dissipates. And then when they looked at the epigenetic markers next to it, and they look particularly at SET7, which is an histone methyltransferases, they had also the same result. The level will increase all the time after normoglycemia. The same thing apply for the methylation of the histone 3. So what do these studies teach us, or what is the conclusion of them? We finally reached, at least at some times, and we can argue about the diabetes study which have been done in the past 2-3 years. So there has been a name as a conclusion of those studies, which we all call the metabolic memory. So it's given to the phenomenon whereby previous exposure to metabolic perturbations has long-lasting pathophysiological effect after the event has dissipated. So, as I just mentioned, several experimental studies have demonstrated the implication of epigenetic in the metabolic memory of culture cells exposed to diabetic conditions. These cells continue to express pro-inflammatory and fibrotic genes long after restoration of normoglycemia. What is the striking and the important thing in here is the metabolic memory is a phenomenon many believe to be responsible for the irreversibility of cardiovascular complication. However, this might be a subject of debate, and I'd be glad to talk with anybody after the session regarding if that is correct or not. So, in order to examine this metabolic memory, my team and I did some retrospective studies here at Weill Cornell with the collaboration of our colleagues at Ahmad Medical Corporation. So we looked at the methylome of patients with type 2 diabetes and high cardiovascular risk or established cardiovascular disease. We looked at the whole range of HbA1c, and the most important, that patients with a bad glycemic control were offered intensification of their treatment, and epigenetics were assessed afterwards. However, we did not study whole blood cells for two reasons, although it could be much more easier to study whole blood cells. We studied monocytes because monocytes are always considered somehow the surrogate endpoints of cardiovascular complications. As we all know as cardiologists, monocytes infiltrate the endothelial membrane. They take the lipids to the foam cells and so on. And also in a lot of microvascular complications of the diabetes, such as the diabetic nephropathy, monocytes are also implicated. They become active macrophages, and somehow they secrete the profibrotic genes, and diabetic nephropathy progresses. So microcytes are somehow implicated in many complications. And the second most important thing why we did not take whole blood cells but only monocyte is what I just mentioned earlier. There have been a lot of studies that suggest that each cell has its own epigenetic signature. I mean, with respect to other colleagues that always assess the whole white blood cells, it might be correct to do it when we're studying about GWAS, but it's not the case when we're studying epigenetics. So this is a brief flow chart of the study. Patients were divided. It's a very easy study. In non-diabetics, diabetics and good glycemic control, diabetics with very bad control. And so each group was defined by a palier of 1.5% of HB1AC. And then finally, patients with more than 10, all patients with an HB1AC more than 8.5 and 3 cardiovascular risk factors or a previous cardiovascular event were invited to enter the interventional study. The inclusion and exclusion criteria are very much typical of a diabetic cardiovascular study. We just excluded pathologies that are known to play a role in epigenetics, such as chronic inflammatory diseases and cancer and so on. So I will briefly mention about the method and analysis. It's a lot of bioinformatic tools have been used. Just to go easy with that, so first of all, monocyte, we isolated them through cell sorting after staining the desired population, and we did the enhanced reduced representation bisulfite sequencing. In other words, a lot of genetic studies are done via chips. So what we did is we really sequenced the gene for the methylation marker. And the way we did the analysis is we divided them into methylation per CpG and methylation per gene. So we looked at the genes, so where the methylation, and we also looked at the CpG, and so what are the genes next to them that could be regulated. So these are like the baseline characteristics of the patients. The patients were already matched for age, sex, and gender. As you can see here, they are pretty much well balanced. Surprisingly, when we compare non-diabetics to diabetics, non-diabetics have a higher prevalence of hypertension and dyslipidemia, and among patients with diabetes, the one with a very bad HB1AC have up to 48% prevalence of retinopathy, which is not really surprising when you have so bad diabetes control. Then when we look at other markers, as you can see in here, the glomerular filtration rate is pretty much decreased in patients with diabetes as compared to control. The total cholesterol, however, the HDL and the LDL are higher in patients with diabetes. In non-diabetic patients. This is probably because of the other one that are taking much more statins. Eventually, also patients with diabetes are much more on aspirin, whether for primary or secondary prevention. And patients with diabetes are taking more ACE inhibitors and ARBs. So let's skip directly from the bioinformatic analysis to this. So we were able to identify 223 differentially methylated genes between non-diabetics and diabetics. However, as I just mentioned earlier in the slide, there are a lot of differences between both groups, and those differences have to be taken into account for. So after correction for all of those differences, we had 115 differentially methylated genes. So just reporting genes is not really something interesting, but we had to look at the function of those genes. And to our surprise, those genes were not just diverse like this, but they belonged to networks. And those networks are either already well known, although the mechanism was not very much known, or they are like networks that we were able to see in the string database with no function at all. So I'll just give you one example of this. This is an important network that has been described in PLOS Genetic in 2012. It's called the mitochondria insulin network, which is involved in the crosstalk between the mitochondria and the insulin signaling system. As you can see in here, I mean, I think there might be something wrong. So the methylation targeted the ETHE1 gene, the TST gene, and the GCT1 gene. So all of these were in the same network. In the previous literature, these genes were known to have gene fusion and to be co-expressed. So what we're saying here, or what our data is mentioning, that methylation could be responsible for doing for all of this network working together and changing the mitochondria insulin network. Skip the other one just because of the sake of the time. And the second thing we did was to look at the gene expression and do some functional validation. So the first thing, we looked how many genes are able to predict the gene expression of the same gene. And we got only 24. However, among these 24 were genes already known in either diabetes or cardiovascular disease, such as the cardiomyopathy and the obesity, the renal failure. But we are a bit disappointed that out of those genes, and we had only 24 out of 115. So what we did further is not only to look at the specific gene, if it's regulated or not, but we looked also at the networks of genes together. And this is when we got surprised that these 115 genes that we identified were responsible for over 15,000 gene expression, which is a really high number, which is higher than the number of genes that can be expressed. However, one gene can be regulated by a different gene. So let's take the example of the SLC 25A5. This is a gene, type 2 candidate gene that has been described in PLOS Genetic in 2012. So when we looked at the methylation of this gene, the expression of all those genes with it was changed by only one gene. So this is something that takes a lot of analysis and a lot of validation, and we're working on the rest of them. And the third category is when the gene methylation of one gene works on the expression of another gene which does not have any function. And this is an example of the ETS1 gene that is important in heart fetal development and vascular inflammation. However, the rest of the other genes were not regulated, and only genes that work separately have been regulated. And we're working right now on the validation of the rest of the network. So I will, in conclusion, so epigenetic changes related to obesity are actually observed in monocytes if we use these monocytes as surrogate endpoints of cardiovascular disease in patients with high risk or already established disease. The changes mainly target network of genes with a specific or a similar function, and the methylation changes do not target the gene expression of the same genes, but extend to the network of genes with a similar function. And what we are doing right now, and hopefully we are at the end of it, so maybe next GHA meeting you'll have many more interesting results. Can we predict the diabetes complication only by the DNA methylation? And the last thing, will the epigenetic changes, I'm sorry for the typo, will they be able to change once we improve the glycemic control? And I would like finally to acknowledge all the funding that I'm receiving, whether internally or externally, and thank all of my collaborators at Weill Cornell, whether in our campus in Doha, New York, Ithaca, and my partners in here at HMC and CIDRA who helped a lot in recruiting the patients. So thank you very much, and I'm looking forward to your questions.
Thank you, Sherbet. Any questions for Dr. Abi Khalid? Maybe I'll start with one. So you mentioned monocytes and why you chose them. Monocytes have a very short half-life, so 2 days in circulation, and then they either move to tissues and they differentiate to macrophages. So the epigenetic changes must have happened before their differentiation to monocytes. What are your thoughts on that?
Yeah, thank you for the question. It's a tricky one, actually. Well, I think that before their life ends, and before new ones get differentiated, they are already imprinting the epigenetic marks. There have been recent reports to suggest also that epigenetics can also be transmitted. And it's happening through generation and generation, not only acquired and then we lose it after we're dead, but can also be acquired. So I think the monocytes are still getting this from the mother or stem cells. From the other part, when you have the cardiovascular complication, and the monocytes are already circulating, they're already programmed to have like this. And there have been a lot of diabetes studies where they show as soon as the monocytes go into the circulation, the polarization changes pretty much, and they're already programmed for the diabetes disease.
Any other questions? Yeah, Mark. So the question is, how about in the field of progenitor cells?
It's a very good thought. We thought about it. It's just maybe for some logistic reasons we didn't do it. And actually, it's not a secret that we've been working with Dr. Mashaka's team and something like this. So I think this will be our next step. But in this study, we did not look at it.
Okay, thank you, Chairman.
Thank you.
So last but not least, we have a presentation by Dr. Samer Kabani, who's an associate professor of medicine at the Lebanese American University in Lebanon. He's an interventional cardiologist with tons of clinical experience and runs a large clinical service at Rafiq Hariri Hospital in Beirut. Dr. Kabani has extensive expertise in clinical work, and he's actually an entrepreneur as well. He started a company called Smart Science, which is a CRO that works in the domain of cardiovascular disorders and metabolic disorders. And actually, Samer and I were old college classmates, so it's a great pleasure to have him here. Samer.
Thank you, Khaled, for the introduction. And I'm going to try to be brief because it's the last talk. And I'm not going to go into all these details, inflammatory process and diabetes and so on. It's just going to be gross information about clinical trials. We're not going to be talking about any basic science. It's all about clinical trials, and mainly in the MENA region. So what I will start is there's nothing to disclose regarding to this talk. And this is the outline of my talk. I'll be talking about the globalization of clinical trials. Why should it be a global trial rather than certain countries? I'll talk about clinical research in the MENA region, what's going on in the MENA region. I'm going to talk also about cardiovascular and diabetes research also in the region. We're going to also go over challenges in the MENA region about clinical trials. Do we have enough clinical trials in the MENA region? What are the opportunities to enhance the clinical trials in the MENA region? And are there any solutions for this region, hoping to improve the clinical trials? So why should a clinical trial be a global trial, not in a certain country? First of all, there's a need of diverse patient population. If you do a clinical trial in 1 or 2 countries, you will not get really a diverse population. And when the medication gets on board and is approved, it has to be delivered to all these countries. So you have to test these medications on a certain population. Definitely, if you do a global trial, it's going to be faster. So the trial will end much faster. The cost will be much less. there is also a chance to have access to rare diseases. If you only do a clinical trial in 1 or 2 countries, it's very hard to collect patients with rare diseases. So globalization of clinical trials is essential. And definitely on a global form, there's demands for medication globally. You're not going to release your medication in the countries you're going to do clinical trials. This medication will be released globally all over the world. And treatment definitely should be tested in a population with potential use. So it is inappropriate to do a clinical trial in one country and to sell the medication in another country. Clinical trials has to be done in the population that you're going to sell the medications in. And as we see in 1990, there is less than 15% of clinical trials were conducted outside the United States. But as things progresses, recently more than 30% of clinical trials are being conducted outside the US, mainly in Central and Eastern Europe, in Russia, India, and China. But what's lagging is the MENA region. We're still lagging a lot on clinical trials. Very small numbers are being conducted in the MENA region. If you look at this graph and you look at the population of the US, it's around 300 million, and around 30% of the clinical trials they run in the United States. If you look at the population in the EU, around 500 million people, and around 30% of the clinical trials also run in the EU. If you go down to the MENA region, Middle East and North Africa, the population is almost 440 million individual, much more than the United States, and it's only 1.4% of the clinical trials run in the MENA region. So the difference is huge. And I think there's a lot of work should be done for the MENA region to enhance the clinical trial operation in that area. This is a recent data from the clinicaltrial.gov where they record basically all the data or all the clinical trials that is occurring right now. In the United States, it is registered 100,000, over 100,000 clinical trials being operated in the US. In Europe, over 74,000 clinical trials being operated in Europe. If you go down to the Middle East and North Africa, or Africa, it's very small number. It's 10,000 in the Middle East, 6,000 in Africa. So if we go down a little bit more details, this is Africa, and you see who's doing research in Africa, or who's doing the clinical trials, mainly in Egypt, around 2,000, and in South Africa, 2,500. The rest of Africa, basically not much of clinical research is happening. How about the Middle East region? If you look at the Middle East region, Lebanon, what's registered is 350. Down south of Lebanon is 6,450, which is a huge difference. If you go down to Qatar, is 94. United Arab Emirates, it's 161. And in the KSA is 515. And Oman is 33. So there is still difference in culture, difference in clinical trials, even among the Mediterranean area here. So now if you look at just diabetes and cardiovascular disease in the MENA region, it's not all trials. When you look at the clinicaltrial.gov, you can know, or you can tell in Qatar, there is around 11 studies being done on diabetes and cardiovascular disease out of the 94. These are the active and recruiting, not the done, that already done. So there are several studies on diabetes, several studies on cardiovascular diseases, and the total is 11. If you look in Lebanon, actually, currently is only 3. And I was surprised when I looked at the data. It's bad. So currently only 3. Maybe there are some being done, but it's not registered at the clinicaltrial.gov. But what is registered is only 3. In Kingdom of Saudi Arabia, what they registered for diabetes and cardiovascular disease is 26. Now the question is, why is that? Why is the MENA region is lacking behind, and what are the challenges in the MENA region? So if you look in different countries of the MENA region, there is lack of education on clinical trials and research methodology. So it's, when you go into a university degree, nobody teaches clinical research, biostatistics, methodology. Very few university settings will introduce these into the trainee, basically. So there are limited, definitely what you will have are limited numbers of publication by the MENA postgraduate trainee, very limited number of basic science research, and very limited number of investigator-initiated trials. So there's not much training and not much operation. And definitely the main reason you can tell there is limited resources and funds for investigators. So it's not like in the EU or in the United States there's much resources and funding. In the MENA region, the resources are very limited and they are not organized. So other challenges in the MENA region, also the current series of crises and political instability, this will give negative impact on the region image, and it's very hard to bring any clinical trial from Europe or from the United States to be implemented in the MENA region. There is shortage of trained professional staff that can support principal investigators, nurses, research coordinator, pharmacists. And there is a need for constant and careful monitoring of the regulatory changes in various countries. So these MENA region countries, they sometimes update their regulatory process and they try to follow the European or the American standards. And the quality of data and the credibility of the data entered also sometimes is questioned. So global pharmaceutical companies usually are worried about the quality of data. And definitely in each country you will need logistics for a clinical trial materials to be imported, stored, distributed into that country. More challenges is the regulatory requirement. The time takes for an import license to be imported into the MENA region sometimes exceeds 3 months, and this delays the operation of clinical trial. And IRB approvals also. Sometimes IRB approval takes 3 months or 6 months to get an IRB approval from a university or a research center. This also discourages CROs and pharmaceutical companies to bring the clinical trial to that country. Also, there should be, to ensure the study is conducted in accordance to GCP, and time and financial investment for that trial monitoring can be discouraging the pharma industry to conduct clinical trials in the MENA region. So these are mainly challenges that needs to be overcome in the region. Now, are there any opportunities? Is there anything which is benefit from the MENA region, not only difficulties? Definitely there are opportunities. First of all, the Middle East and North Africa region, you have around 400 million individuals. So this is a lot of burden of disease and a lot of patients that can be recruited, and the annual rate of growth is 1.8%, which is compared to 1.2% of the global population growth. So people are basically growing in this population. And definitely there is demand for medication in this region. Well, the demand has surged a lot. And the disease pool in the MENA region sometimes differ from the European and from the American. It's typically the diabetes. The incidence of diabetes is much higher in our region. There's also genetic and cultural differences in the MENA region, different than the European or the US. And there is a high rate of consanguineous marriage and geographic isolation of subpopulation that contribute to a distinct genetic pool. So in certain population you have different diseases, different genetic pools. And there's definitely different metabolic abnormalities in the MENA region, especially obesity and diabetes. And there is a high prevalence of inherited blood disorders that are not present in the European areas. And we know there is a high death rate that are related to cardiovascular disease, cancer, and diabetes. So this is also looking at the prevalence of overweight, obesity, and diabetes in the MENA region countries, and you can see almost 70% of the population are overweight. There is almost 30% of the population are obese, and diabetes almost reaching 20% of the population are diabetic. So these are really serious diseases that need to be taken care of. Moreover, the percentage of total death in cardiovascular disease, cancer, and diabetes really leads the causes of death in the MENA region. So opportunities for expansion for clinical trials in this region, we have young population, and most are clinical research naive. They haven't been enrolled in any clinical trials, which is very important to get naive patients to try new treatments on these patients. And there is a cultural difference where there is a trust between patients and their physicians. And this will help a high enrollment rate and help a high retention rate, rather than especially for the long-term clinical trials. And definitely in the MENA region, we have good research sites, we have experienced investigators that needs basically to be enrolled in these clinical trials. We have good academic centers affiliated with international hospitals and definitely affiliated with US, and they are following US and European standards. And moreover, about opportunities, if you look at this numbers, this is a density of patients recruited in clinical trials in accordance to in a million population. So in North America, for every million individuals, there is 100 individuals recruited in a clinical trial. In Western Europe, for every 1 million individual, there is 65 patients recruited in a clinical trial. In Australia and New Zealand, for every million individual, there is 58 patients recruited in a clinical trial. However, if you go down to MENA region, for every million individual, there is only less than a person recruited in a clinical trial. So there is a lot of individuals, a lot of diseases, but not much people are enrolled in clinical trials. So are there any solutions to enhance the clinical trials in the MENA region? For sure, and we have to start from the basics, basically. You have to start enhancing training of new graduates on methodology of research. So university sites, university centers, they should put programs to encourage people to go into research, basic science and clinical research. Definitely we need to improve the funding for basic science research. We need to improve the collaboration with other countries and academic centers in other regions. And we need to further involve physicians and investigators with phase 2, phase 3, and phase 4 global clinical trials. Now, definitely to be able to get involved in clinical trials, you need to have clinical research units. And there are specific criteria to build up a research unit that accept clinical trial according to EU standards or US standards. You have to have sites with basic infrastructures that is required. You need the principal investigators who are interested in conducting clinical trials and have some experience. You need to have a research coordinator to support the principal investigator. You need your unit to have a collaboration with your pharmacy department and the laboratory services. And you need to have a greater use of a centralized institutional review board. So if a study comes into a country, the sponsor doesn't have to wait month and month for every center to get an IRB approval. And definitely you have to have adequate financial funds for all staff involved in the clinical trial, so they will do their work and they will finish their work as quickly as possible. So a clinical research unit requirement, you need a working space for people to come in and work. You need a research unit administrator, so human resources is very important. You need a research coordinator to support the investigator. You need to have a phlebotomy service that supports you in your clinical trial. You need also to have a pharmacy service, and the pharmacist has to be trained how to take care of investigational medicinal products. You have to have an archiving space because all these documents will be archived for at least 15 years at the end of the trial. And definitely you have to have an IRB, and the IRB has to have a relatively quicker or a quick turnover time. And definitely you have to have standard operating procedures for every single step in the conduct of clinical trial. And you need to set up your pricing for any clinical trial, and you need to know how to negotiate the contract. And I'm going to give you some ideas about contracts and what are the pricing that it should be in the contract. You should negotiate about your PI fee, sub-investigator fee. There is definitely an IRB fee and an IRB renewal fee. Every time or every year you renew your IRB approval, you can charge your sponsor. There is a startup fee. There should be an import license fee, a SUSER's reporting fee, archiving fee, research coordinator fee, a pharmacy fee, a laboratory and a phlebotomy fee, a radiology, EKG, and echocardiography if needed, a closeout fee, and definitely a clinical research unit overhead that covers the indirect cost. So these all should be in your contract, and definitely if you run a clinical trial and you're able to collect all these, this is a fund that you can put it in a box and you can use it for investigator-initiated clinical trials to support your residents, your fellows, and to support further enhancement of your research department. So I'm gonna give you some success stories of a research unit that we developed in Lebanon, and even Lebanon is a very small country, but you can see how much achievement it did, with a population of 5 million in Lebanon, not much. So this is a study that we did, and this is one of the success story. Actually, we did almost over the last, let's say, 5 years, over 70 clinical trials in that unit. I'm just gonna give you some issues that we have got some of the success stories. And this was written to us. With your dedication and huge work, we have reached on 13 February 2013 our targeted number of patients to be randomized for the TAU study. The TAU study was an acute coronary syndrome sponsored by Sanofi, very difficult study, patients coming into the emergency room and directly you have to start them on a new anticoagulant, and we were blinded whether the patient is receiving heparin and a 2B3A or this new anticoagulant. The patient goes to the cath lab, he do the procedures, and we follow them after the procedures. And they were extremely grateful and would like to sincerely thank you for your contribution. Congratulations. Total of 53 patients randomized in Lebanon, exceeding the target for Lebanon by 13 patients. So they gave us a certain target and we were able to basically exceed the target. Another achievement for a clinical trial that was run globally by Novartis, globally in the US, in Europe, in Australia, and what we basically were able to achieve, once again, we want to give our special praise to our 3 top enrollers that for 2 consecutive months are leading enrollment with more than 12 patients per month. China was the first enroller, Lebanon was the second enroller, and India was the third enroller, and the rest of the world was down actually. So we're beating the US, we were beating EU, we were beating Australia. Basically just we couldn't beat China. So even though we got also an award from a clinical research site award from INC, INC really is a big global CRO that operates clinical trials worldwide. They came and they gave us award at Rafiq Hariri University Hospital, CRU. So basically, irrespective the size of the country, irrespective the number of population, if you are dedicated and you have a dedicated team, you can achieve really big time. So there are 2 principles for clinical research. Clinical research should be conducted in a population in proportion to the potential uses of products after approval. So it does not make sense to do research in one country or one region and sell the drug in another region. And in the Declaration of Helsinki expresses an expectation that every patient enrolled in a clinical trial should at the end of the trial be assured access to the best proven therapy identified in the study. So we should not leave our patients after a clinical trial that if the drug has been approved, he should receive the drug and until it is approved in your country. So I will stop here and I'll thank you for inviting me, and I'm here to take any questions.
Good evening. Thank you for the talk. It was excellent. Have you been successful at establishing a collaborative relationship with any academic centers back in the U.S. or in the EU?
Actually, yes, we are, but honestly, it's a little bit tricky. I'm a graduate of the Cleveland Clinic, actually, and I could not establish something with the Cleveland Clinic up till now. But we have some collaboration, but it's relatively weak. But this is really, we keep meeting with them, we keep organizing with them. We have a good relationship with University of Montreal, Montreal Heart Institute, and we're running some of their clinical trials in Lebanon. And definitely other fields, I'm talking about cardiology, but other fields, oncology and endocrinology, have a good relationship with other AROs, and they are trying to bring as much as they can clinical trials into Lebanon.
So I have a quick question. In terms of the regulatory framework that you've touched on to encourage research, so one thing we've been talking about in Qatar for a few years now is potentially establishing a national IRB. So that will entice collaborations between different hospital systems and so on. Anything similar in Lebanon or the rest of the MENA region that would enhance this type? I mean, like you said, the regulatory framework is one of the biggest hurdles toward conducting research. Anything like that happening?
No, in Lebanon, actually, each hospital has its own IRB. We try to establish a central IRB, but it was very difficult because I believe of the culture of people and the difficulty in case something happened to the patient, who's going to take responsibility and so on. But the trick is trying to select individuals from each institution and create an IRB, and each individual will reflect or will support his institution. And this definitely will enhance the sponsor to be able to bring clinical trials into that country, because they don't want to invest for one site or two sites. That's a big investment, and they will get one or two patients. If you have 10 sites, then they will consider the country. And if you tell them our IRB turnover or approval, it will be within a month or two, definitely they will be interested.
Thank you for your talk, Samer. I've got a question regarding, I can see somewhere in the Middle Eastern countries and in Lebanon in particular, there are a lot of IRBs that are like not affiliated to university hospitals, when we're talking about the private CRO. So who regulates this IRB? Who controls it? How does it work?
Usually IRB should be regulated by the Ministry of Health. They put SOPs, and then to have an approved IRB, you send your application to the Ministry of Health. The Ministry of Health review your SOPs and review the criteria that they did put it. And if they agree, they will send you a letter that this IRB has been approved by the Ministry of Health, and it is allowed to go ahead and approve clinical trials. In Lebanon, we have the Ministry of Health that has set criteria for certain IRBs, and every hospital wants to do a research. They will form their IRB committee, they write their SOPs, they send it to the Ministry of Health, the Ministry of Health reviewed it. If they agree on that or they have any changes, they will send them to make the changes, and they will give you approval for a couple of years, and they will post it on the website. So not every research unit in Lebanon or every hospital has an approved IRB. And you cannot do clinical trial in any hospital in Lebanon. You can do it in selected sites in Lebanon, which they have approved IRB by the MOH.
Great. Thank you again, Samer. Let's take a moment, thank all the speakers for a wonderful session, and thank you all for joining us tonight.